There we are, we're recording. Okay. Hello everyone, welcome to the next episode of my science podcast. Today I am delighted to welcome Dr. Greg Rouse, who is a professor of marine biology and curator of one of the Scripps biological collections. His research entails animal phylogeny, biodiversity and taxonomy, particularly in annelids, echinoderms and seadragons, as well as the evolution of whalefall, hydrothermal vents and methane seep fauna. We are also I'm proud to also welcome Avery Hatch, who is currently a graduate student researching into and understanding the evolution and biodiversity of branchiate scale worms. So thank you so much both for joining me today.
Thanks for inviting us, Martha.
So may you start off by giving us an overview of both of your careers and why you entered biology as a profession?
I'll start and then hand it over to Avery. So I'm originally Australian, but now work in San Diego at the University of California, San Diego. I've been here about 15 years, and I moved here from Australia because of the deep sea research opportunities that are available in the United States compared with Australia. But I did all my training in Australia for the most part. My father was in the Navy and we traveled around a lot in Australia and Asia. So that really introduced me to my dad introduced me to the marine world on the weekends. And decided I'd like to pursue marine biology when I was fairly young, about 12 or 13. I remember getting a book as a prize in school that was one of Jacques Cousteau's Underwater World books, and that really helped further inspire me. So I then started a Bachelor of Science in Zoology at the University of Queensland, and then went on and finished a PhD at the University of Sydney. I then was lucky enough to get a postdoctoral fellowship at the Smithsonian in Washington, and my mentor there was the world expert on polychaete annelids, and I worked well with him for a few years, and that really kick-started me with the opportunity to stay in marine biology. And so then I went back to Australia and had several jobs there before being recruited here to Scripps, and I've been here ever since, and run a lab with a number of graduate students, as well as curating our benthic invertebrate collection. And Avery happens to be one of my students.
Hello, thanks so much for inviting me today. So my name's Avery. I have always been in love with the ocean. So when I was 7 years old, repetitively I went with my dad and stepmom to the Monterey Bay Aquarium. So we had memberships back then. So every time I'd visit them and spend the summers with them, that was like our family activity. We'd always go. One time we even got to sleep over at the aquarium. So I had a lot of fond memories, and I was really, really big on writing in my journal. So I still have journals from when I was 7 years old. And I have a specific page where I wrote about my stepmom told me what a marine biologist does, and I decided that's what I want to do. So ever since then, I mean, that's exactly what I wanted to do. So I came to UCSD originally with the intention of wanting to be a graduate student at Scripps. But then once I graduated, I realized I wanted to learn more about the field and understand what it really looks like in real life to be a scientist instead of just taking courses.
Yeah. Okay, so Greg, what are the phylogenetic and biogeographical implications of the diversity of branchipollino and that you discovered from methane seeps and hydrothermal vents?
So this is actually a good prelude to Avery's question, and that one of the things we really do in my lab is explore these amazing ecosystems in the deep sea where there's a lot of energy coming out of the crust in terms of volcanoes, which heat up water in the surrounding sediments, and they blast this water out, superheated water at 350 Celsius, and it gets saturated with various chemicals, including hydrogen sulfide. And hydrogen sulfide can be used by a variety of bacteria, and then various animals graze on the bacteria or have incorporated them as symbionts into their bodies. And these include various kinds of mollusks and as well as various kinds of annelids to highlight the major players. This also occurs at methane seeps, where usually along continental margins of the world, you have methane associated with petroleum deposits or other events that are geological, such as subduction, where continents are sliding over each other. And this methane comes out of the sediments and often in large quantities, and is also a big energy source for microbes that can break it down, often resulting in more hydrogen sulfide than the same community of bacteria and animals can exploit. But the diversity varies from place to place. And sometimes animals can overlap at seeps and vents, but often we find different communities in different places. And so we're interested in the evolution of those particular communities. And we focus mainly in the Eastern Pacific because that's where we've had most of our cruises. Hydrothermal vents were first discovered off the Galapagos Islands, where there's some continental splitting going on in terms of a famous place called the East Pacific Rise is nearby, and there's a Galapagos Rift. And the methane seeps were first discovered in Florida in 1984 actually, and then methane seeps have now been discovered in many places around the world. So we've been fortunate enough to sample methane seeps all the way up from Oregon down past Costa Rica, and hydrothermal vents from Galapagos through to off Oregon, as well as in the Western Pacific. So the mussels, which are actually closely related to the shallow water mussels that people like to eat, have co-opted bacteria into their gills. There's not a lot of energy in terms of filter feeding opportunities for mussels in the deep sea. So they can often be found around seeps and vents using bacteria they're basically farming the bacteria in their gills to give them energy. And there is a very unusual group of scale worms called Branchipolynoe that live inside the mussels as symbionts and potentially as parasites. And these are quite big worms. The mussels can be 15 centimeters or so long, and the worms can be several centimeters long, quite large. And often there's a pair of them living inside the mussels. They're very bright red, very beautiful worms. The red comes from hemoglobin. They're living in a very low oxygen environment, and they have lots of these branching gills coming off their bodies. And so they're really adapted to tolerating low oxygen environments. Remember, they're living inside a mussel that's mostly closed, so they have no fresh water going over them, and often the water is low in oxygen anyway. So we found 4 new species at Costa Rica methane seeps and integrated their evolutionary biology into an overall phylogeny of the group from the Atlantic and also the Eastern Pacific hydrothermal vents and Western Pacific vents, and really showed an interesting pattern in the evolution of these seep and vent organisms. Sorry, just a minute.
For me?
Absolutely. So may you tell us about how these branchiate scale worms are adapted to thrive in hypoxic habitats and survive toxic concentrations of chemicals like hydrogen sulfide?
Okay, so Greg kind of mentioned a little bit about the adaptation of the branchie, but just to begin with, so branchiate scale worms, the term branchie is referring to the gills. So they have very elaborate, well-developed gills underneath the elytra. So elytra are another term for their scales, which are the beautiful large scales overlapping the dorsum of the body. So these gills have been known to really help with diffusion distances. So they minimize the diffusion distance for oxygen, and they have a large surface area. So specifically, besides just the branchie, and all of the genera that have branchie have been found in the deep sea, mainly at chemostatic environments specifically. As for the toxic concentrations of chemicals, not too much really known yet. There's a lot of research to be done. There's one study, however, that was led by Yanji Zhang in 2017, and they studied comparative transcriptomics. So this is a type of science that studies the transcriptome and compares them of different species in order to understand adaptations to the environment. So it makes the assumption that differences between the transcriptomes may reflect to specific traits of the environment of that animal. So I'm not sure how well versed you are with genetics in general, but it seems like you did a lot of background research on our work. So the transcript, just to clarify, is the genes in the region of cells that you're extracting that are being expressed at that given time. So instead of just all the information in the genome, you're specifically targeting which genes are being expressed. So it's really useful to see which genes are used for that animal in that specific environment. So this study looked at the transcriptomes of 2 deep sea species and 1 shallow water pollinoid, which is also a scale worm. And the main findings that they found were that the adaptation to hypoxia in the genome, which they were able to see, was the evolution of tetradomain hemoglobin and also a high expression of single domain hemoglobin. So hemoglobins are what bind oxygen. So they showed a way higher saturation of expressing these genes than in the shallow water species. But as for the toxic concentration of chemicals, not too much is known yet. And also we're still studying the ultrastructure of the branchiae. So there's only really been one study that looked at the group of branchipollinoe, so the composition and ultrastructure of those gills, the same worm that Greg was discussing that lives in the mussels. So Greg and I are actually interested in looking at the different types of branchiae that are seen across all the branchiate scale worms, because it appears from our evolutionary tree so far that they may have evolved several times. So we're interested in looking at if they have the same type of branchiae or if they're different, because most of them have, 4 of the genera have a very similar appearance with like arborescent tree branching. So they have like long finger-like projections, and there's 2 batches of them. So there's a large surface area. And the study that looked into the branchipollinoe had found that they are actually filled with coelomic fluid that has extracellular hemoglobin, which creates the presence of the red pigment in the animals. So that's what we know about those branchiae, but we're really interested in looking more into the branchiae and how they're adapted in the other species, the chemosynthetic.
And so how did you use mitochondrial COI or 16S rRNA and nuclear 18S rRNA or 28S rRNA and H3 genes to reveal the 4 new species that Greg mentioned before in the Pacific Ocean?
I think Greg was referring to the branchipollinoe species for that first question, but the Painilea pollinoe species So I extracted DNA from different Painilea pollinoe species, and then I used basic Sanger sequencing to do PCR and sequence and target those 6 specific genes. So I'm not sure if you know much about PCR, but polymerase chain reaction. So essentially what I did was I had different sets of primers. So you need a forward and a reverse primer. This DNA is read from the 5' to the 3' end. So the forward primer is designed to bind on to the 5' end, and the reverse primer binds onto the 3'. And DNA polymerase, the enzyme that naturally exists in our cells, is used to replicate the DNA. So by creating this reaction and having different annealing temperatures, which is the temperature that allows the primers to bind specifically at that temperature. So by creating a thermal cycler program, you can amplify the DNA over and over and over again exponentially by repeating these cycles. So you have a cycle that denatures the DNA and splits up the double strands. So that's like at high heat, like at 95 degrees. And then you have the annealing temperature, which allows the primers to bind to all the little snippets of DNA where you want to target. And then you have the elongation temperature, which is actually the temperature that allows DNA polymerase to start replicating that specific sequence that you're targeting. So that's the technique that I use to sequence those 6 genes. And then in order to check if you amplified the correct regions, you use gel electrophoresis. And that is a technique down when applied with, and based on the weight of the DNA. So if the DNA is heavier, it moves less after a given amount of time. But if the DNA fragments are very short, it'll move faster. So essentially what you want to see is if you had amplified the region that you're looking for with hundreds and hundreds and hundreds of copies, you're going to want to see a band at that designated base pair length on the agrostele. So that's how you check your work to make sure that you targeted the correct region. And all of the rest of the genes in the genome are kind of not gone away. They still exist in the DNA extraction, but they're not highlighted because all of it is concentrated into that one amplicon. So after doing that, then I Sanger sequenced the product and I aligned all of the different sets of genes and then I concatenated them together. So essentially what that means is you take each gene separately and you align it with all the others of that species. And you do so for each gene, but then you don't want to create a phylogeny just based on one of the genes because you want as much data as you can get. As Greg later will talk about, the more genes, the better resolution in the phylogeny. So what you want to do then is kind of combine the alignment side by side to have more data, the better. So I did that. And essentially, we found new terminals in the phylogeny amongst the greater group of deep sea scale worms. And we assessed the genetic distances between the other Panulae polynoe species that were already described. And we decided that they were new species based on the genetic distances, based on the phylogeny, and based on morphological investigation.
That's absolutely fascinating, and a lot of that detail will be really brilliant for a lot of our listeners. And so on a similar genetic level, Greg, what technology allowed you to discover the mitochondrial genome of the freshwater annelid Manayunkia occidentalis? Sorry if I pronounced that wrong.
No, that's correct. We are transitioning in many ways from what Avery was describing, which is called Sanger sequencing, named after an Englishman, actually, Sanger, who invented the technology to target a particular gene and amplify it many, many times to allow us to sequence it. Nowadays, we use a different sequencing technology. It's often referred to as next generation sequencing, although it actually is several different technologies bundled under the one name. Another term for it is often shotgun sequencing. You're just randomly sequencing parts of the genome. So we are starting to do that routinely because it's become very cost effective. To get a single fragment of a gene using Sanger sequencing costs us about $8 to $10 and per specimen and per gene. So we can, though, for about $100 now, do what's called genome skimming, where we just randomly skim sequence parts of the genome. We'll do an extraction of the DNA from an organism and then ask the company to give us random pieces of DNA from the genome to a certain level that you pay for. We know then that out of that random DNA, there will be a mixture of mitochondrial DNA fragments, which has its own mitochondria have their own DNA genome, which is about 18,000 bases long, so it's relatively small. And then the nuclear DNA, the genome varies according to the organism, and it can be very, very large in terms of gigabases down to hundreds of megabases. So the scaling of this is a part of the decision making as to how much sequencing you do. Nevertheless, we expect about 1 to 2% of the data we get back to be mitochondrial DNA, and that allows us to actually reconstruct the entire mitochondrial genome. And so rather than paying to do Sanger sequencing, we now are tending to invest for $100, we'll get the whole mitochondrial genome, and then if we need to get some nuclear data, we will do that. We also utilize another technology called transcriptomics to get data as well, which isn't really associated with that question that you're asking me. The Maniunkia occidentalis genome, though, was actually done by a collaborator on the paper that you're referring to, and they did the whole genome effectively with a lot of coverage to the level where they could properly document the nuclear genome, but they hadn't gone in and pulled out the mitochondrial genome. So Ekin, who was working in my lab at that time, pulled out that mitochondrial genome from Maniunkia from the entire genomic data that was available, and we then published that. We also used that in conjunction with describing Maniunkia occidentalis as a new species. Maniunkia was originally described from the eastern US, and occidentalis is Latin for western. So we showed then there was a new Maniunkia species here in the western rivers of the United States. And this is actually important because Maniunkia is a host for a parasite that is primary host is salmon. It gives them a disease called whirling disease. And there's a lot of interest in Maniunkia as this intermediate host for this parasite that's having a lot of economic effects. And we showed actually it wasn't the western species was new, it wasn't the eastern species, and that's going to shake up how people understand the ecology and distribution of these parasites.
Absolutely. And so my next question is on your work on leafy sea dragons. So how has post-glacial flooding with rising sea levels produced locally variable signatures in colonizing populations of these leafy sea dragons?
Yeah, so sea dragons are a great interest of mine. So I'll have to give a bit of background, I think, before I can answer that question. There are 3 species of sea dragons. They only live along the southern coast of Australia. And I was working on them before I came here. Well, I was working on 2 of them before I came here. At that point, there were only 2 known. And part of our research led to the discovery of the 3rd sea dragon, the ruby sea dragon, in less than 5 years ago. And you can read about that online if you want to find out. That was with another PhD student of mine, Josephine Stiller, leading that work. But our original interest was actually in the genetics of sea dragons. And we were concerned about their conservation status. It was unknown in terms of ICUN guidelines. And these are iconic fish along the southern Australian coast, very favoured by scuba divers, and they're very popular in aquarium exhibits around the world. We knew that no breeding was happening in aquaria. No one had really cracked breeding of these fish in captivity. And so the fish were being harvested routinely from the wild. And we just wanted to get a snapshot of, one, whether there was just the 2 species along the southern Australian coast, because often you get eastern and western species in Australia of fishes. But the argument was that the weedy sea dragon was occurring all the way from Perth in the west, around Tasmania in the east, and up to Sydney and north, if you know your Australian geography. So all along the southern coast, whereas the leafy sea dragon was more restricted from South Australia in the middle of the southern continent, and then all the way west to around Perth. So we did mitochondrial genome data and some nuclear data on these, and published that early on a few years ago, and drew some inferences. First of all, that there were just the 2 species, the weedy and the leafy species, with a side project proving the ruby sea dragon as a 3rd species. But there wasn't a lot of power in that mitochondrial data to draw inferences, and we then adopted another kind of next generation sequencing technology, focusing on something called ultra conserved elements. And these are little snippets of DNA where our DNA, for instance, would be identical among all humans for this little snippet, but also would be identical even if we compared it to a fish and to a frog. I mean, they're amazingly conserved little regions of DNA. And so these can be targeted and used, because they do vary a little bit, for making evolutionary trees. But we decided to do something novel, which was to actually do sequencing outside the edges of the UCEs to find the more variable regions and use those for population genetics. And this is a very cost-effective method, and we were able to scale up our understanding of the genetics of sea dragons by using well over 1,000 gene fragments. So this was much more powerful than what we had done before. And it allowed inferences at a very localized spatial scale, and also allowed us to have a look back into the past, relatively recent past of the sea dragon history. And this then allowed us to start to understand what had happened with sea dragons in terms of sea level changes. So as I'm sure you know, Martha, we've had about the last 2 million years of ice ages where we've had cold and warm periods. We're currently in what's called an interglacial now, but as little as 12,000 years ago, the sea level was much, much lower than today, about 120 meters lower, because we were in an ice age, and the glaciers and polar caps were much, much larger than today. And so sea levels were correspondingly much, much lower. Now, in places that have a big continental shelf, we saw the continents expand very greatly and go much, much further out to sea. And this is the case for, say, the US eastern coast, which goes out for many, many miles, very, very shallow. However, off Australia, the continental shelf is pretty narrow. And so if you drop the sea level by 120 meters, you get to the continental edge and very deep water very quickly. Sea dragons live in shallow water for the most part, in kelp forests. And so if we saw a major drop in sea level, they actually ran out of habitat. And so what we think happened repeatedly with ice ages is sea dragons had their available habitat shrink dramatically, and then they were restricted to relatively small regions of the Australian coast. And we see this in the genetic signatures of the population today. And we were able to show that in the West, the genetic diversity of leafy sea dragons really went through a bottleneck and hasn't been able to expand. And in the South, though, where there were some large embayments, which were dry, they did flood again, and leafy sea dragon populations expanded into those. So we get greater genetic diversity in leafy sea dragons in the central south of Australia, and they were then able to recolonize in the west, where the sea dragon habitat had really become limited. So this paper was recently published in Molecular Ecology, and really shows the power of this next generation sequencing and allowing really nuanced studies even at a local and relatively recent time period.
Definitely. And so how could conservation actions focus on preserving genetic diversity at local scales and maintaining habitat connectivity for these leafy sea dragons?
So we're concerned with both the leafy and the weedy sea dragon. And so we have another big study coming out on the weedy sea dragon on their genetics, and that will then really inform the different states in Australia. So sea dragons are all protected at a federal level, but the states have their different protections in place for the sea dragons. And we really want to be able to give them a more nuanced view in terms of particular populations that might be isolated and should get special attention, and maybe have no collecting or any behavior by divers. They might need special looking after. And so our data will provide that information. We're also building on that genetic work and we have been running now for some months, a project called Sea Dragon Search, where rather than resampling the populations to study them for genetics, we are now doing citizen science approach, where people who take photographs of sea dragons can upload them to our website, and we can use automated learning, artificial intelligence, machine learning technology to identify the sea dragons to individual. So we have a website, seadragonsearch.org. We work with a company, Wild Me, with their technology Wild Book, when they've used this on whales, sharks, cheetahs, zebras, lots of other animals, and sea dragons are one of their few fish projects. And we're getting lots and lots of data now on individual populations of sea dragons that will mesh very nicely with our genetic data as well. So check out the website. It's very cool. Even if you live in a place that doesn't have sea dragons, there's a lot you can learn about them through our website, plus just see the amazing pictures of these animals.
Absolutely, and hopefully a listener might join in and help with your research. And so, Avery, the sparkling iridescence of the Phenylleopolynoorphinae appears to be due to a thickened cuticle of the scales, and so for protective rather than communication purposes. How do you believe this iridescence has evolved?
So before going into that direct question, I'm going to give you a little bit of background on this story and this finding because it was very exciting for us. So for years, since I started looking at the Phenylleopolynoe diversity in general in about 2014, we noticed that there were several different color morph variations. And at first we thought potentially maybe there were different species because the color forms were very drastic. So there were representatives that were red, some that were white, blue, pink, and there was only one specimen that we found that was black. So due to that, we became curious in the first place of what were the mechanisms of these different colors and such. And in addition to that, when we had collected several samples, looking at them, a lot of the time, the larger individuals would have like destroyed parts of the scales, kind of looked like rigid edges ripped off. And we thought maybe this could be due to collection process because a lot of the time animals get damaged when being brought up to the surface. So we weren't really sure what it was. And then Greg actually in 2018 in November saw them live and got ROV footage of the worms actually fighting one another. So they have these evertable proboscis that the pharynx comes out of the body, and at the tip of it, they have these jaws. So they were using their jaws to bite off chunks of one another's scales. So it's pretty remarkable. I don't know if you've seen the YouTube video, but I'll have Greg share that with you if you haven't.
I'll link this to the podcast.
Oh, perfect. So that is what gave us the insight that these actually were bite marks in the elytra. And knowing that, looking back at the variation of specimens, it seemed that none of the smaller individuals, the juveniles, seemed to have these destroyed marks in the elytra. And also, on top of that, all of the small individuals were the colors that were either white, a light red, but all of the largest individuals, the most mature, were always notably blue and pink and very, very iridescent. So we seem to think that maybe the scales were thickening with age, possibly, and the idea of the protection came from the clue of seeing the fighting occur in situ. So because of that, that's where we kind of came up with that idea to begin with, that they're most likely protective, and possibly the cuticle of the elytra thickens with age. However, we do not know how they evolved this iridescence because in the deep sea itself, there is no light. And these specific worms also don't have eyes. So they don't use the iridescence as like an attraction to one another or for signaling, because there would be no function for that in the deep sea. So we think that the iridescence is possibly a byproduct of the ultrastructure of the scale itself. So currently we're actually starting to finally look into the composition of the elytra and also the ultrastructure between the different color morphs to see what changes with the thicker blue and pink ones and what is creating this iridescence. So we don't necessarily think that it was selected for throughout evolution, but it came along with the selection of thick scales for protection. So yeah, we'll find out more on that soon, but we're currently collaborating on a study looking into this with Sylvia Vignolini and her team, and also Dr. Ekin Tillich.
Wonderful. And so back to Greg. May you describe the symbiosis between the two species of annelid and distinct aerobic methane oxidizing bacteria belonging to the methylococcales localized to the host respiratory crown?
So here's a collaboration that it was led by Shana Goffridi. And Shana is a professor at Occidental College in Los Angeles or Pasadena. And Shana and I have worked together for many years, initially on the bone-eating worms that take out, initially whale bones was the first discovery, and we'll talk about those in a little bit, I think. But whenever Shana and I are on cruises together, she's always on the lookout for novel symbioses between animals and bacteria. And I'm like the animal guy, so I'm interested in the biodiversity. And then if we see something unusual that looks like it might have a potential new symbiosis, then Shana will take the lead on that. So I've talked about the various kinds of animals that have symbioses of hydrothermal vents and methane seeps, such as mussels, and there are other kinds of clams, and there are gastropod snails that show this. And among the annelid worms, there are the most famous ones are giant tube worms that are called vestimentifera. And these can be several meters long. They're the iconic organisms you see at images of hydrothermal vents from the Eastern Pacific. And there are also some other ones, though, that have bacteria living on their skins. And one day, in a cruise where we were in the Gulf of California, and also where we were in off Costa Rica, there were some very unusual worms that I had studied a lot. They're called fan worms or feather duster worms, that the anterior body, the head, is transformed into a filter-feeding crown. And the body of the worm is hidden inside a tube. So you don't really think of them as looking like a worm at all until you take out the tube, take them out of the tube. So here's a filter feeder living in the deep sea, where I've already said with mussels, they don't have much opportunity to filter feed. So that was the first suggestion that something strange was going on. There often is a bit of flock, like snow, bacterial snow around, and we were thinking maybe the feather duster worms could be filter feeding. But the feathery plumes themselves looked strangely like they had a mat on them. They looked a little unusual when they came up and in the video. So Shana then took some of these crowns, these feathery crowns of these worms, and she sequenced the crowns, what was randomly sequencing them for whatever bacteria was on the crowns, and also then took the crowns and embedded them for studying the tissues, and did that in a particular way where she could also label fluorescently any bacteria that were on the surface of the crowns. And she showed then that with DNA sequencing, and then also with the fluorescent technology, and then we also did the ultrastructure to show that there were bacteria all over these feathery crowns, and that they were methane oxidizing bacteria. And this was particularly at Costa Rica, there's methane coming out of the seafloor, and that's what's driving the ecosystem. So the bacteria were not living inside the feather duster worms, they were living on the surface of the crown. And there was definitely evidence also in what is called stable isotope analysis that the worms were utilizing these methane oxidizing bacteria. There's a particular isotope signal in consuming methane oxidizers. So somehow, in a way that we don't properly understand as yet, because the bacteria were living on the surface of the animal rather than in the animal, so there was no direct way that they could consume them. They are definitely providing nutrition, though, to the worms. And this was in two different groups of feather duster worms. So one of them we've now named, the two species, they're species of Lamina tubus, and that's an open access paper that's now been published. And then the other one is in another group of feather worms called sabellids, and that one we still haven't published, and we will in the future. But it was a fascinating new discovery of symbiosis, and Shana also just recently led another new one where sea anemones are also using these methane oxidizing bacteria for their nutrition, living around hydrothermal vents and seeps also in the Gulf of California and Costa Rica.
Absolutely. And so you mentioned this before, but how do Ozidax use symbiotic bacteria, proton pumps, and carbonic anhydrase to secrete acid in order to dissolve the bone carbonate matrix to then allow the absorption of bone-derived nutrients across the skin?
Yeah, Ozidax is an animal a worm. It's an annelid worm, but it looks, when you see it on the surface of the bone, and this is only occurring in the ocean. So this happens on whales and other vertebrates that have died and fallen to the seafloor, and all of their flesh is stripped away, exposing their bones. And in 2002, Bob Reinhoek at Monterey Bay Aquarium and Research Institute was leading a cruise and found a dead whale on the seafloor at 3,000 meters, but it looked like a red carpet. It was completely covered in these red, beautiful flower-like creatures. And Bob sent some of those to me. Meanwhile, they were doing the DNA work, and the anatomy of these creatures was very strange. The flower-like structures went into the bone and then branched through the bone like the roots of a plant, and every one was different. All of these worms were females, and the large part of the body inside the bone was actually an ovary. And then the red plume was actually feathery crown that was full of blood vessels. So, and the red color came from hemoglobin. So the worm's roots were full of bacteria. And so here was another example of symbiosis, but in this case, they weren't going after methane or hydrogen sulfide. They were actually bacteria that specialized in breaking down organic compounds. And we subsequently established that what they were after was the collagen. Our bones have a large collagen matrix around which the apatite, the calcium phosphate matrix, is then deposited. So the worms were going after this collagen, but that meant they had to dissolve away the matrix. Whale bone, and any bone, as you know, is very hard. So they use a mechanism on the skin of their roots to create an acidic environment to dissolve away the bone. And this was work done with my collaborator, Martin Tresgueras, here at Scripps, and he's a physiologist. And so we did experiments where we were able to isolate and show the activity of certain proteins that were working inside and outside the worm. The roots showed very high activity of a particular protein, VHA, which is something that we have as well as humans that is helping to pump protons and create an acidic environment in our stomach. These worms were doing the same thing out of their skin and creating an acidic environment in the bone and dissolving away the bone. They then had to get rid of that matrix, and here's where the carbonic anhydrase comes in. And so there's a whole physiology in terms of creating an acidic environment, but then they still need to get rid of the dissolved substrate as well, and they're pumping it out through their crowns. This then let them have access to the collagen, and in a mechanism that's not fully understood, because collagen's a big molecule, they had to get that collagen broken down and through their skin, because the bacteria are living well inside the body of the worms. We do know from isotope ratios that we've done before that the worm isotope ratios and then actual lipid chemistry that the worms are living off the bacteria. So there's definitely a relationship then between that the food pathway is whale to bacteria to worm. And so we've got a basic understanding of how these worms are able to exploit bones. And it's a fascinating study in terms of their biology, reproduction, evolutionary biology, and it's one of our major research focuses in my lab.
Absolutely. And so what did you find when investigating the nervous system of these Ozidax?
When you look at Ozidax, these females, they don't look like an annelid worm at all. Annelids, the most familiar one you might know of are earthworms that have a series of ringed segments running along their bodies. And so most annelids show this repetition of segments, and Ozidax didn't show that. It had a relatively smooth body. It had different regions, but no obvious segmentation. And so we wanted to look at the nervous system to try and understand if there was any trace of segments. And what Ozidax is also very strange in having is mostly there are females that are dissolving the bone that don't look segmented at all. And then they have these little male dwarfs that live in the tubes of the females. So they have dwarf males. the males do show segmentation. So we were comparing the males with the females, and we were able to find some basic regionalization of the females in terms of what was a head compared with normal annelids and what was a body compared with normal annelids. But there really was very little trace of obvious segmentation. Whereas in the males, we can clearly show the presence of a couple of different segments. And this was done using immunolabeling with again, similar to what was done with immunolabeling DNA, we can immunolabel proteins. We did this with the VHA in the skin of the worms. We can do it with a nerve indicator like serotonin, or a nerve indicator like tubulin, which is a very common component of nerve fibers. And this makes the nervous system light up under fluorescence imaging, and all of the other tissues you don't see at all. So you can trace out the nerve pathways. So that was a fun project that's still letting us get a handle on Ozidax anatomy, which we're still exploring from other aspects as well.
Yeah, that's fascinating. So my final question is back to Avery. Can you tell us what other projects you're doing for your PhD?
Yes, I'd love to talk about all the projects I'm working on. So currently, right now, I am working on assembling the full mitochondrial genomes of several different representatives within a monophyletic clade that we have determined called Lepidinotapodini. So it's a subfamily of scale worms that are all found to be native at chemosynthetic environments. So these species are found at whale falls, other types of organic falls like other wood fallen in the deep sea and bone, also methane seeps and hydrothermal vents. So I am looking into investigating the mitochondrial genomes because a study led by Zhang, published in 2018, had shown that there is a fascinating amount of gene rearrangement occurring in the mitochondrial genomes. So typically, a lot of annelids actually have conserved mitochondrial gene order, but for some reason, a lot of these deep sea scaleworm species are showing lots of different gene orders. So the mitochondrial genome has 37 genes total. It has 22 tRNAs and 13 protein coding genes and 2 ribosomal RNAs. So I'm investigating the orders of those genes, and I'm also using the large 15 genes to create a stronger phylogeny of the group. So I'm interested in understanding the evolutionary relationships better and the diversity of the animals, and also there's lots of new species. So after this research of looking into the mitogenomes and hammering in the phylogeny to make some taxonomic revisions, I'm going to be describing 8 new species within that clade that live at chemosynthetic environments.
Wow.
So they don't all have branchiae. This monophyletic clade contains pollinoids with and without branchiae, but according to our phylogeny so far, it does look like branchiae has evolved more than once. There are 2 main types of branchiae that we see just by looking at them without deep investigation. So there's the arborescent tree branching branchiae that we've been talking about. And there's also one species in a monotypic genus that has plicate branchiae. So they're kind of folded. They're folded over. So they're flat, but they have a large surface area, kind of like the concept of the surface of our brain. So there's those 2 different types, but also within the arborescent branchiae, there are different positionings of them. So we're curious to look into, as I mentioned before, if the actual ultrastructure and the composition and the function of the different branchiae amongst that clade are similar to the Branchipollinoe that's been studied, or if they're a different type of branchiae. So that's another project we're looking into, the new species descriptions, the mitochondrial genomes, and then ultimately I really am interested in comparative transcriptomics as well, which I mentioned there was some light work that's been done so far. So I want to get more samples and collect deep sea specimens of Lepidinotapodini, as well as other deep sea scaleworms that don't live at chemosynthetic environments. So these are specifically from the clade that is sister to the Lepidinotapodini clade. So they're the closest ancestors. and they're found at seamounts. Some of them are found at chemosynthetic environments, but it's not as largely seen as in Lepidonotapodini. So I'm interested in comparing those two different types of deep sea transcriptomes as well as shallow water taxa.
That's absolutely amazing. And it really highlights how unexplored the deep sea is, and it's the biggest habitat, as Greg said at the start, but it's so unexplored and so much more needs to be done because it's fascinating. And so the work that you're both doing is brilliant, and hopefully my generation will bounce off it. And yeah, you're doing amazing work. So thank you so much for joining me today. Do you have anything else you'd like to talk about before we end it?
Well, I can certainly tell you there's plenty more to do, and the ocean is a wonderfully unexplored place. It is suffering from some threats right now, but don't be too down about it disappearing. There's still plenty to explore and plenty to see, and the more interest we have from people like you, Martha, who then choose to go on to careers in ocean science like Avery is doing now, the better the chance we have to understand and conserve it.
Absolutely. Avery, do you have anything else you'd like to say?
Not particularly. I got a little derailed earlier with the baby. So I was kind of anxious trying to give my introduction, but I did want to mention one of the highlights of my experience was during my undergraduate degree in 2014 in the spring. I was a student in one of Dr. Greg Rouse's classes, marine invertebrates. So it was a laboratory class, and we got to always do microscope work and draw animals and investigate the anatomy and the function and learn about taxonomy. And it was fascinating. I remember it was like a long class, like 6 or 7 hours twice a week. And everyone, like a ton of the students would be like out when it was like the class was done. And I would always be looking around like, oh, everyone's gone, still drawing. So I knew I loved it. And I would go home and spend time fixing my drawings just because I wanted to make them better. And it didn't really matter for the course itself. So that kind of really triggered my interest in specifically marine invertebrates. So I kind of went from like a broad idea my whole life of knowing like I want to study the ocean, but I didn't really know what I wanted to do. That's why I didn't jump into a graduate degree, because I was like, what is out there? What does a real career look like doing this? Because I had no idea. I knew I liked everything, but I knew that that's not feasible. And I knew that I wanted to be able to investigate and know deeper knowledge on a specific subject. So I asked Greg if I could volunteer with him. So that's kind of how my journey into the Rouse Lab began. And I volunteered for a couple years, 1 or 2 years. And then I was working part-time as the lab technician. And then when I graduated, I was really grateful to receive the offer to work full-time as the lab manager and lab technician. So I did that for 2 years. And I knew towards the end of that, I'd really made up the decision in my head, like I want to be a graduate student. I was looking around at all the other graduate students in our lab, and I was so jealous that they got to spend time on their own research. I was like, that is such an honor, and that is so cool to go home and be able to spend all this time on your work and take pride in it and find out more about it than anyone's ever known on the subject before. And that just fascinated me. And Greg took me in my first like large fieldwork expedition in October of 2018. I was super, super honored, and I got to go with him to Costa Rica to study the methane seeps off of the coast there. And he granted me the huge honor of going in Alvin, the submersible, with him.
Oh, wow.
So that was a really triggering moment in my career too, and that really, really meant the world to me. So all of those experiences kind of culminated into me deciding I wanted to be a graduate student. And at first, I wasn't really sure where. And then Greg had mentioned on our way home from the airport on that trip, you know, you could do a PhD with us. And I thought about it as like, why would I ever want to go anywhere else? I was like researching into labs, and I knew I loved genetics. and I started getting into bioinformatics and computer coding, and I was addicted to it. So I was looking into other labs that did that sort of thing, and I was like, they're not all nearly as cool as the lab I'm in. What am I doing? Yes, I want to be a student in this lab. So that's definitely, I think, one of the experiences I wanted to be able to wrap it up with, of how I specifically got into Greg's lab. And I never knew I'd be studying the deep sea, but now I'm just fascinated by it. And there's such a plethora of new animals and so much to discover about the new animals now that we know. Like, it's so unfortunate that so many of the questions you ask, I'm like, I don't know, but what we do know, and everything I want to know about, I feel like there's no answers to yet, which is really exciting. And
it is exciting.
But thank you for inviting me.
No, it's been wonderful. Thank you so much for joining us. And I'll say thank you everyone for listening, and I'll see you next time.